So now you see the presentation, right?
Yep, that looks good.
Yes.
Okay, perfect. Um, all right, let's give this a shot. So welcome to the workshop. This is going to be a workshop on dimensionality reduction and visualization. And also, it's a really ambitious title. We're basically going to talk about like what these things are that we do. So if you open up then, or sorry, have people managed to load up the repo as well? Just want to make sure.
Yeah, it's loaded for me too.
Okay, yeah. So like you managed to click the binder button and it all worked.
Yep.
Okay. Uh, all right. So the purpose of this workshop. So I'm going to try and give some intuition on dimensionality reduction, clustering, visualization, things like that. Um, why you might or might not use different methods. And the methods that I'm going to talk about are principal component analysis, PCA. So this is sort of the workhorse of dimensionality reduction. UMAP, uniform manifold approximation and projection, and also T-distributed stochastic neighbor embedding, or TSNE. So the latter two methods are quite closely related, but they're very common these days in either human genomic work, but especially in like single cell work. And I'm sure if you've read those papers, you've probably seen these types of embeddings. I'm going to give you some resources to read and some code to play with. So this is in the repos, or in the repo in the notebooks. You'll see a lot of it there. And the notebooks are interactive and walk you through a lot of the code. So you can play around with them after this workshop, and email me if you have any questions. But yeah, there's a lot of real data in there. There's toy data. Um, yeah, in terms of the resources, I worked hard on these notebooks. I do recommend you read them because we're not going to be able to cover all the content in this. Oh, sorry, this should be 2025, because I ran into some sort of like library issues. Uh, yes, hours of fun, and the notebooks are set up so that the introduction has uh, explanations of the intuition plus some useful papers, some of which I've written. So, I always feel proud when people read my stuff. There is one notebook for toy examples, which covers artificial or example data, a genotyping notebook. So this covers the 1,000 genomes data. And there's also a ScanPy notebook, which uses single cell data. And there's also an R pack, or there's also an R chunk of code, which uses the Seurat package. So if you're more of an R user, you can work through that as well. So I won't have time to cover that one, but that's also included in the repo. So yeah, you know what? I'm just going to change this right now because I feel like if I ask people to remember things, it's not going to work as well as just manually changing it. So there we go. That's the new repo. All right, back into this. Sorry, I don't know why my keynote is doing this. All right, I'm going to hack this a bit because I have notes that I'm going to read off on my second monitor, and you're just going to look at this. I hope this is okay for everyone. Um, but yeah, so why would we want to do dimensionality reduction? The basic idea is we want to take our data from a large number of dimensions to a low number of dimensions. So like in the most obvious example, or one of the most common examples is actually, let's say you're working with the globe. The globe is in 3 dimensions, but we want to look at a map because it's way easier to look at a map and carry it in your pocket or look at a screen. Now, maps are in 2 dimensions. So this is a really, really common example of dimension reduction or dimensionality reduction. I have it in parentheses. I switch between the two all the time. I'm just going to say dimension because it has fewer syllables. Um, but it's a really common motivating example for dimension reduction, where you might want to get an idea of what an object can look like, but in a simpler format. So, like shadows are another example of dimension reduction. Shadows exist in 2 dimensions, but obviously the objects that we see are in 3 dimensions. Now, if you look at a shadow, it'll give you a good idea of what something looks like, depending on the light source, what the angle is, things like that. But you can also imagine a shadow where the light source is not pointed well might hide a lot of information. So you just example like imagine like you see a table, and if you have the shadow that goes straight down from the table, you just see a rectangle and you wouldn't get any information. Whereas if you have one that's kind of from an angle, you could get an idea of like, oh, it's got 4 legs, they're kind of connected by a plane, that sort of thing. So these are multiple representations that you have of the same data in low dimensions. But thinking about the globe to map example, the globe is a sphere and maps are planes. And we actually can't represent that data perfectly in 2 dimensions. So if you imagine peeling a globe the way you would peel an orange, and kind of flattening out all of the globe, you're going to have to tear the map in some way, or you're going to have to stretch the map in some way. These are sacrifices that you have to make when you're going from a high dimensional space to a low dimensional space. And if any of you are familiar with different types of maps, you may know that like there's the Mercator projection, or there's other types of projections like a polar projection. But each of these projections preserves a different aspect of the data. You know, if you're looking at a nautical map, maybe you want to preserve oceans more. If you live in the United States, maybe you want the US to be more prominent in the map, so you use a projection that emphasizes that. But you will always have to sacrifice something, which is why Greenland looks like the size of Africa on the Mercator projection, even though I think it's, I don't know, like a tenth or the 20th the size of the continent. So that's a really common motivating example. Uh, but yeah, the idea is you also want to preserve important aspects of the data. So what is important in your data? That's a question you might want to ask. PCA is really popular for a whole bunch of reasons, but one of which is that it maximizes the amount of variance explained. So if you wanted to have the lowest dimensional representation of your data that explains the maximum amount of variance, PCA is actually the perfect one for this. And this is because it is made up of the eigenvalues and the eigenvectors of the covariance matrix. It is by design the explanation of the variance in your data. Um, One of the things it's not so good at though is like maybe you have really, really local structure. So you might have a bunch of cells or a bunch of humans and say they're closely related or similar to one another. You might look for clumps of data specifically, like clusters. And you might want to emphasize neighborhoods and local structure in your data a little bit more. So methods like UMAP and TSNE are really good for this. The drawback of TSNE and UMAP is you're going to have to distort your data by breaking the long distance relationships. Um, and then you might have other questions like, say, you want to ensure continuity or time scales, like if you're working with embryonic data and you have single cell data from different time points, or like tumor data or something like that. You may want to have, make sure that like, oh, I want the cells from like day 2 or day 3 to be closer to the ones to day 0, and I want them to sort of maintain this flow. So there's methods that work on like heat diffusion models. So FATE is one of those. It's not as common as the others, but I see it here and there for that type of data. But, in terms of why reducing dimensions in the first place, so there's kind of two paradigms in statistics that I like to think about. And on the left, this is what's called, and you can see my cursor, right? Like if someone can give me a nod. Yeah, okay. So on the left here, we have what's called Fisher's paradigm. And then on the right, this is Tukey's paradigm. So the idea in biology, say 150 years ago, well, let's say 100 years ago, was that you had fairly linear hypotheses of your data. The idea was, okay, you have a question like, does this corn grow taller than this other corn? And you would develop a hypothesis, and that would be your null hypothesis, your H naught. You would design an experiment around this hypothesis. You would collect the data. You would compute a p-value, and you'd get your conclusion. It would be pretty straightforward. We're kind of in the opposite situation right now, where often we're working with data that we haven't collected, or we have collected the data, but it's so unbelievably complex that we can't just start with a simple hypothesis and work with that. So John Tukey, who is a statistician who published a lot between the 60s and 80s, he came up with a different paradigm, which is where you start with the data, and it's more data driven, and you visualize it. And the idea behind Tukey's paradigm is that, okay, there are formal ways to do statistical tests, but one of the first things you should do in the scientific method is observe your data. But how do we observe it? Well, we have to visualize it. So we make some sort of choice about how to present it. We will create graphs or scatter plots or something like that. And then we'll look for forms. So things that kind of stand out like clusters or really elongated stretches of data, maybe outliers. And you have a bunch of options there. You can say, okay, I have a simple hypothesis. Maybe I have like clusters of cell data, or I have really unusual outliers in my biobank. You can say maybe there's something special about that. Maybe it's a batch effect. Maybe it's a measurement error thing. So you can either create a hypothesis around it, you can do a simple diagnostic, and you kind of go through this iterative process where maybe you need to collect more data, or you can say, you know what, I think I have an idea of what I'm going to do. I'm going to start doing confirmatory analyses of the data. And then you can either collect more data or you can go to a conclusion. So those are kind of the two paradigms that are highlighted in a lot of biological statistics. And this is a figure from a textbook from Holmes and Huber, and this is listed in the introductory notebook that I have there. But it's a really great notebook if you just want a textbook on statistics, like modern statistics for modern biology. So that might be the title of the textbook. If you're looking for some examples of dimension reduction, I wrote a review paper with a bunch of other very cool people that I met in 2024. And it's an overview of the motivations of the motivations of why you would do dimension reduction. Also, some of the philosophy behind it, because there are arguments back and forth. People might say like, "Oh, my method's better," or "All these methods are bad. Never use TSNE, only use PCA," things like that. But there's a bunch of different arguments that go back and forth, and this covers some of them. But it also gives a survey of all the methods, all the applications, all the limitations, and I think importantly, it shows a whole bunch of different examples of different types of data. And something I really want to get across here is that all dimensionality reduction is it's all different representations of the same data. So you're going to be working with the same data and you have to make choices of what it is that you want to preserve in your data. And when you visualize it, when you cluster it, when you do whatever, you need to keep these assumptions in the back of your mind at all times because this will influence how you do your analyses. And this is the group, so there's me hanging out in the back, squinting because it was super sunny. But yeah, this is the developer of UMAP here, developer of TSNE, developers of Fate. So we did basically a survey of how common all the different dimensionality reduction methods are. So TSNE, you can see, is still really popular. UMAP was developed in 2018 and started to take off. PCA is excluded from here because it's just so old that it doesn't have a canonical citation. But I mean, if we included PCA, everything else would look like a sliver because PCA is used like everywhere all the time. Um, but these are some of the examples of things you can do with the data. So the notebook, one of the notebooks I've provided, actually works with 1,000 genomes data. Self-organizing maps. SOM, is that what you meant? I just see there's a question in the chat. Yes. I mean, they're still commonly used in courses. I haven't personally seen an application of SOMs in a long time. Back in my master's degree, I played around with them a bit, but as far as I know, they haven't really been used very often. It may have specific applications, um, but to be honest, the main application I've seen is like you want to create a map, but you only have distances, and you don't necessarily want to put anything in there. So it's still used, but I think at this point it's I don't know if outdated is the right word. It's just not super common that I've seen in biology, at least in a long time. Um, so the 1,000 Genomes Project, this is just a bunch of genotype data, but so a PCA kind of creates this classical triangle shape because it maximizes the amount of variance explained in the data. And generally, the maximum variance in human genetic data tends to be related to geographic distance. So you can see that when you label these by the continental populations, the terminals of the data, they tend to correspond to the continents. Laplacian eigenmaps just makes three little dots. TSNE, you kind of get this fuzzy, cloudy structure, but you can see that the clusters, so the colors here, by the way, I should mention, are the populations from the 1,000 genomes, but you can see that they kind of form these cloudy clusters. MDS, which is multidimensional scaling, forms sort of this circle pattern. Fate tries to keep this continuity between them, and then UMAP over here has like much tighter clusters compared to TSNE, but sort of preserves the continental structure and sort of preserves the local substructure as well. Um, so this is another example that we have, which is mouse cortex cells. So you can see similarly, PCA tends to really stretch out. So this is from the Tazik et al. Paper in 2018, and the colors are transcriptomic cell types. But you can just see that the different representations of the same data highlight different aspects of it. So PCA, you kind of see clusters within it, but it kind of shows where the data tends to vary. TSNE forms these clusters. UMAP also forms similar clusters, as does Fate, but they have varying degrees of continuity connecting the clusters themselves. There's an example of primate brain organoids. Um, and you can see sort of the same thing. So here with Fate, we have the sort of continuous preservation of the data, and this is because you have temporal data, so it was designed to kind of keep this ordered. One of the drawbacks of a method like TSNE or UMAP is the clusters will often break or tear, and that is because they focus on preserving local neighborhoods in the data, as opposed to PCA, which explains variance, and fate, which tries to keep continuity between them. So this is something to really consider with a method like TSNE or UMAP. You will have artificial tears between the data. So if you think about the map example, you do have to tear it in some place or another based on the method that you're using. UMAP and TSNE, because you're saying I only care about like the neighborhood that's nearby, it might say, okay, well, the long distance is not meaningful. Um, and then, just to show a totally different type of data, we have Wikipedia data. So this is just a bunch of paragraphs from the English language simple Wikipedia, about 500,000 paragraphs of text. And you can see that these others, so I'll talk about these four here in the corner. They kind of form this big cloudy structure, and you can see, okay, Wikipedia is roughly like, some pages are more similar to another. Um, but when you look at methods like TSNE or UMAP, you get these really clear clusters that actually correspond to topics. So, you can see all the music tends to cluster together, all the movies tend to cluster together, things like that. Um, so just to go back to the Thousand Genomes one, because I'm a little bit more familiar with this, why is dimension reduction necessary in the first place? So typically when you're doing a statistical analysis, you have P, and all this that I'm discussing right now, this is in the introductory notebook. You have P parameters, and let's say these are genetic variants, single nucleotide polymorphism, SNPs, whatever. But this also holds for like cells. And you have N samples. When you're working with a lot of genomic data, you're often talking about, you might have like a million SNPs. You might have 20,000 gene markers, and you might only have a few hundred or a few thousand individuals. Or you could be in a situation where you have, say, 500,000 individuals and millions of SNPs. You need to do dimension reduction in order to do anything with the data at this point, because otherwise you're going to run into a whole bunch of problems. Like visualizing that data is pretty much impossible. Clustering it is going to grind your machine to a halt. And yeah, any sort of statistical analysis, you really need to bring the number of parameters down lower. So yeah, a really common example in modern biology is defining cell types. So traditionally, you might look at a cell under a microscope. So we go to the transcriptomic example here. You might look at a cell under a microscope and say, okay, we're going to talk about the size, the shape, the color, and then we'll try to classify these cells. This is really labor intensive, and it is sort of arbitrary since you could have really different cell types that happen to look similar. But what if you wanted to work with genetic data and look at something that was more intrinsic? So let's say you had measurements of proteins or DNA or RNA or something like that, and you could classify the cells this way. What you could do is get all of these gene measurements from, say, 20,000 genes, but it's really hard to figure out how to kind of bring them together in one way or another. Um, Now, we know lots of genes are highly co-expressed in different cell types. So a method like UMAP or TSNE or or PCA, you can actually just run the method that you want, create a visualization that you find convenient for the properties that you're trying to preserve, and then color it in by the label that you have, by some gene expression, and then that'll give you a more intuitive idea that's more data driven as opposed to just kind of looking at it and trying to figure out like, I think this cell kind of looks like this other one. Maybe they're kind of the cell type. So there is always going to be an element of subjectivity, but at least this is a little bit more data driven subjectivity. And there's tons of examples that I've seen in papers for finding examples or finding patterns looking at brain scans across voxel data. You might look at gene expression across embryonic data, subtypes of cancer, using cell surface protein measurements, looking at admixture patterns across populations, finding patterns in microbiome distributions, and clustering species or subspecies, or you might just want to make your data more efficient for some downstream process. Um, So that kind of covers the examples for now. So these are some things that are really crucial to remember. Dimensionality reduction is always going to require trade-offs. You're going to have to decide what's important in your data. Do you want to preserve neighborhoods? Do you want to preserve variance? Do you want interpretability? Do you want computational efficiency? And you should always keep in mind what is the goal of this dimension reduction. If you're looking for computational efficiency, you just want fewer columns to process. A lot of time PCA is going to be good for that, for denoising. If you want interpretability, Um, I think all the methods are good for this, but they only have a certain number of or they only have certain reliable interpretations, which I'll get to in a moment. And then, downstream processing, clustering, or other machine learning methods, some methods are more suitable than others. Uh, so these are the links, and I can share this presentation through email afterwards. But so this is the link if you haven't clicked on it yet to bring up the repo, and this is what we're using right now. If you want the older repository, this one has some R code in there, so if you go to my GitHub, you can find it. But this is if you're interested in the Seurat package, which we won't be covering today, but there are options for R because this is all going to be in Python. I'm going to pause for a moment here and see if there are questions. Right, there are either no questions or Uh, I've explained everything so well, it just made perfect sense. All right, so I'm just joking. I know there's always questions. So I'm going to share one of the notebooks here. Do people see my Jupyter notebook right now? Yeah, okay.
Yeah.
So I have there's four notebooks in here, and you can see a lot of it kind of covers what I've already talked about. And I'm just going to run through a few quick points that I didn't make it into the slides, but I'll probably skip well, I'll talk about the clustering. There's effectively two types of clustering. We have hard clustering and soft clustering. Hard clustering says like, okay, every point either belongs to a cluster or does not. So this is one where like, you have 10 clusters in your data and every cell, for example, is one of 10 cell types, or it's not any because it didn't go into a cluster. Oh, I see a question in the chat. Okay, bye Brandon, have fun. Um, Soft clustering is also called like fuzzy clustering or probabilistic clustering. This is where every individual point can belong to multiple clusters. So, for example, you might say, so this is really common in population genetics where you're looking at genetic ancestry and you assume there's like, say, five clusters, which are continental populations. And you may say something like, oh, individual A belongs like 70% to the European cluster and 30% to the African cluster. And we would talk about ancestry in those sorts of proportions. There's a whole ton of different clustering methods. K-means is a really common one, but it's kind of computationally inefficient at this point, and it's mostly used in toy examples. So I've got a description in there, Leuven and Leiden, which is community detection, and density clustering. Which is like finding, dense points of data within space. But I have some interesting, or I have a whole paper on density clustering and genomics if you're interested about that. But if you're interested in the philosophy of it, which I think is an important thing to think about, there are two papers here, one by Christian Hennig and one by David Watson. Um, I'll do a brief rundown of the mathematical side, but uh, basically what you're doing is you're just trying to reduce the. Actually, you know what? I'm going to trust you to read the mathematical side of it instead. I'm just going to run through the common methods and talk about their strengths and their weaknesses. PCA, this is the really common one. This is the workhorse of it. So, as I mentioned before, the intrinsic property is that it preserves the maximum amount of variance. So it's got very, very interpretable properties in that every principal component explains a certain amount of the variance in your data. And one of the notebooks hasn't. We'll run through it, but it tells you how to do that. And it also is a linear transformation, so you can do it pretty quickly. The main limitations of PCA is that you may have signal in higher order principal components. Typically, the way you work with PCA is you take your data, you run it through PCA, and you say, okay, we're going to take the top 40 principal components and use that for our analysis. If you have signal in principal component 41, it's going to get thrown away. So that is something to consider. And you can also miss nonlinear relationships in data. So if you have a lot of interesting clusters, they may actually get kind of squashed and forgotten. Uh, a question here oh, this is a really relevant question for me. Oh, sorry, is my sound cutting off?
Sound
No.
was okay for me.
Okay. Sozi might be I don't know if you're on a Bluetooth speaker, because if it's Bluetooth, it might actually be something with battery or connection. Chelsea, I will come back to your question because that is actually relevant to my research. So do remind me before I wrap up so I don't forget.
Okay, thanks.
Yeah. Um, TSNE and UMAP, these are really common methods in single cell and genomics, but they've also just taken off in general. But TSNE is a neighborhood graph method, and the idea is that you basically model the high dimensional data and you try to approximate the distance by a normal distribution, and you model the low dimensional data with a t-distributed statistic, t-distributed probability, rather. And the idea is you optimize the distance between the two of those. So it's really popular because it's very, very good for visualizing data. One of its biggest strengths is generating discrete clusters. So it's good for visualizing clusters, and it preserves the local structure of the data. So points that are near each other in the high dimensional space. So think about like cells that might have similar protein structures, or humans who have similar genetic structure, things like that. They tend to appear together in TSNE visualizations. So it preserves local structure quite well. The limitation, the trade-off that you're making here, you might say, is that long distances in TSNE are not interpretable at all. So if we go back to the presentation I was doing here, um, in PCA, these long distances are very interpretable. You can say that, okay, this distance corresponds to a genetic distance between individuals. The distances in TSNE, like the distance from here over to here, these are not interpretable at all. And it's the same with UMAP. The distance between these clusters here and these clusters here is not at all interpretable. What you can interpret is the local structure, saying the points that are close to each other in the low dimensional space are close to each other in the high dimensional space. However, the ones that are far away, there's no interpretation of that. And that's just a trade-off that we make with this method. And the same goes with the single cell data. So you can say, okay, these types of neurons, I'm not an expert on neurons, but there is an explicit interpretation of this distance between excitatory neurons and non-neurons, and inhibitory neurons. And, it's that the transcriptomic data corresponds to a distance here. So there's a difference that is measurable in the transcriptome here. Whereas in the UMAP or TSNE data, the distances between, say, the far side here and the far side here, these are not interpretable in any way. But you do see these clusters in a much finer scale that makes it really, really useful when you want to think about, like, is there a specific cell subtype? So that's where the strengths of these methods come in. Um, UMAP is very similar to TSNE, in a lot of, like, sort of philosophically similar, but it has a few strengths that to me make it a little more useful than TSNE in some cases. Um, So UMAP, rather than modeling things as a probability distribution in a high dimensional and a low dimensional space, what it measures is as a uniformly distributed data on a manifold. So in the name uniform manifold approximation, that's where it comes from. Um, it's it's similar sort of structurally in terms of having like a slightly different optimization problem, but the main strength that UMAP has is it generates discrete structures a little bit better than TSNE, I find, in that it identifies clusters very, very well. And it can also have a little bit more continuity than TSNE, which is to say that some people would say it preserves global structure better. I don't know if that's the right way to put it, but the idea is, if you zoom in on TSNE here, or TSNE and UMAP, you can see that UMAP does sort of stretch out these tendrils that kind of connect between the different cell types. So the local distances, if you'll recall, are interpretable, but um, excuse me, the local distances are a bit interpretable, but the long distance ones are not. The trade-off with UMAP is that it can be overly discretized. So you see little like floaters over here. These can be totally broken off sometimes. So if you have really sort of really unbalanced population sizes in cells or whatever it is, what UMAP can do is it can actually really pull them away. So if you have a sample of like 10,000 individuals or cells, and there's like 15 really closely related individuals or very similar cells, they might end up floating like way off in the distance, and that's just a consequence of the UMAP optimization algorithm. Everything is kind of a push-pull, like you want to pull things that are similar closer together and push things that are different far apart. UMAP has a very strong push-pull, especially when you have unbalanced population sizes. But In terms of its strengths compared to a method like TSNE, TSNE can really only work in 2D, maybe 3D if you're lucky, but I wouldn't recommend it. UMAP can reduce data to an arbitrary number of dimensions. So if you're doing something like clustering, clustering in 2D is unreliable because you lose a lot of really important relationships in the data. You get a lot of artificial tearing. Clustering in like 5 dimensions is a bit better because those relationships are more likely to be preserved. So you can take your data, run it through UMAP, and then put it in like 5 dimensions and then run clustering downstream. And it works really, really well, especially because of this property where the data is a little overly discretized. You can get really strong clustering. Um, of course, the drawback is your data is very overly discretized. So if you don't want super tight clusters, maybe UMAP is not the right choice, but you can play around with the different parameters in there. Uh, and yeah, it has the same drawback as TSNE where the long distances are not interpretable at all. So like this cluster's distance to this cluster over here is not really relevant. Um, so we can take a moment to pause here before I go to the toy examples. So if people have questions, throw them in the chat, or we can, uh, excuse me, yeah, throw them in the chat. And if they're quick ones, I can address them now, or I can leave them to the end. All right, so there's a notebook in here. It's toy examples. And this one uses toy data. So we're going to run through it and look at PCA, TSNE, and UMAP and how they work with different methods. So there's a whole preamble here that I have on how PCA works. The important part, and I encourage you to read through this and to try and understand it. There's a lot of online sources. Some LLMs are actually pretty good at explaining this as well. Or you can just like email me and we can chat about it for those who work on my floor. Um, but mathematically, PCA works through eigenanalysis. So the idea with, for those who remember linear algebra, eigenanalysis generates eigenvectors and eigenvalues. And if you take an eigenvalue and you divide it by the sum of all the eigenvalues, it tells you how much of the variance that eigenvector corresponding to the eigenvalue explains. So when people talk about, oh, PCA explains a maximum amount of variance or PCA explains a certain amount of variance, what they mean is if you want to figure out how much variance your data explains, you can just take the eigenvalues related to the eigenvectors of PCA and calculate it yourself. And this is really handy because sometimes you can get, you know, imagine you have data with like 100 variables and you run it through PCA and the top 10 PCs explain 92% of the data or something like that. That's really useful because now you've cut your data down by a huge percentage, but you still retain a lot of the variation in the data. So for this reason, PCA is super popular as a denoising step. So a lot of transcriptomic data, for example, they'll run TSNE on it, and they even skip this in the methods, which drives me insane. But usually what they do is they take the top 50 PCs of transcriptomic data, and then they run that through TSNE or UMAP. So one thing you should think about is if I'm running the top 50 PCs, what if there's something in PCs 51 through 100? So for those of you who do work with transcriptomic data and you work with packages like ScanPy or Seurat or whatever is popular these days, uh, keep in mind that if you increase the number of principal components or decrease it, you're going to find a different representation of your data. Because just to hammer this home, you're always working with different representations of the same data. So I really do encourage you to play around with the different number of principal components because you could find some signal even in very well explored data sets that people have just not bothered to look at. Um, but
I
yeah,
have a
just going.
question
Oh
about
yeah,
that.
sure.
Um, because usually you see like PCA plots with the first and second component on a 2D
Mhm.
axis. Are you suggesting instead of plotting the first and second one, like you plot like the third and fourth one on the 2D axis, or do you have to like increase the number of dimensions?
That's a great question. Because one of the strengths of a method like UMAP, which I use all the time, is you can take the top 10 PCs, run it through UMAP, and then it's kind of like getting a summary of the top 10 PCs on a 2D plot. I do encourage people, if you're looking at multiple principal components, don't just look at one versus two. Look at, say, do some pairwise plots, like one versus three, one versus four, two versus five, at least for the top like three or four PCs. That being said, one reason I really like this question is because it highlights the major drawback of PCA in that if people are only looking at the top three PCs, you don't know what's going on in like, say, the top seven, the top 20, the top 50. So what I really like to do is to look at the top PCs, but also like one versus two, two versus three, but also to take, say, the top 10, 20, 30, 40, 50, up to 100 or something like that, run them through UMAP or TSNE and see, excuse me, just have a little water coming up there. Running, say, the top 100 PCs versus the top 50 PCs with transcriptomic data, and then seeing like, oh, there's a whole new cluster here, because that does happen. Because sometimes the signal for a cluster, for like a cell type or something like that, or a subtype, it might not appear until like PC 14. So computation is cheap. Like run a script that just does a whole, maybe does pairwise comparisons like the top five or six. And then when you're working with a method that uses UMAP or TSNE, change the default number of PCs and maybe even change the parameters in UMAP and TSNE, which you can ask me about that later if you'd like. But yeah, definitely examine more than just like the top two PCs.
Okay, thank you.
Yeah. Um, so looking at the toy examples here, yeah. Okay, we can get through this in about 10 minutes. So there's this whole thing of like, what is PCA doing? And I can give you this whole explanation about eigenvalues and eigenvectors. I think the most intuitive way is to say, okay, you've got this point cloud here. So, and can people see this data? If I can get like a thumbs up in the chat. Okay, I see
Yes.
a thumbs up. Good. Yeah. So what PCA does is it takes this point cloud, and this is just randomly generated data. Effectively, what you're doing is you're putting an ellipsoid around it. You're drawing an ellipsoid around it. You're drawing a circle or a stretched out oval around your cloud in whatever number of dimensions that you're doing. And the axis of each of those sort of the longest stretches, each of those corresponds to a principal component. So if you look at this ellipse that I have here, you can see that the data, and this is two-dimensional data, the data is kind of drawn out in this direction. So when you draw an ellipse around it, you'll find that this axis is the longest one. So this is actually going to be our principal component one, and this is our principal component two. So there's there's one thing to consider here. Imagine if instead of this point being here, it was way the heck out here. It was like twice as far away. PCA is very sensitive to outliers because suddenly if you have an outlier that's way out in one dimension, it's going to stretch that ellipsoid way out. So now that principal component is going to stretch way out. And if you think about it intuitively, PCA explains variance. Well, what is variance measuring? Variance is measuring how spread apart your data is. So if you have really spread apart data, your PCA is going to reflect that a lot. Just checking a quick message in the chat here. Oh, yep. See you around. Um, but yeah, so effectively what you're doing with PCA intuitively is you're drawing an oval around your data in a high dimensional space, and then you're creating the axes for each of those for each dimension of the oval. The longest one, the longest stretch of the oval is PC1. It explains the most variation. The second longest one is PC2. It's the second most variation, and so on in that manner. This gets really hard to visualize once you're in dimension like 97, but the principle kind of holds. Um, so obviously it doesn't just draw the oval and the line. What it does is it stretches and rotates the data to transform it. So there's some code in here to illustrate it. But yeah, so this is kind of the intuition behind PCA, and I find this geometry helps me think about it pretty easily. We do have some toy data here. I hope it loads. It does load. Okay, fantastic. So this is the iris data, which is quite famous at this point, but these are a bunch of measurements of different parts of a flower. So there's three species of flower, and it might have actually been Fisher himself who measured this, but the idea is, okay, we have how many dimensions of data here? Length, width for the sepal and petals. That's 4 dimensions. That's hard to visualize. I can only see in three, and it's kind of hard to think in three. I can only like use paper in two. If you can see in more than 3 dimensions, do let me know. I'm still looking for someone like that. Um, but yeah, if you transform your PCA or your iris data into PCA, this is roughly what it looks like. So there's a package that does this for you. You can do this manually through linear algebra, or you can just use like one of the billion libraries out there. So this one just uses sklearn, I think by default, scikit-learn. And this is what the first couple columns of the PCA data look like. So you can see it's quite different because the data has been transformed to something else. So like it's not length or width anymore because you have negative values. But we have four principal components because remember, we had four columns that we were working with, and PCA is going to transform the data. If you're n by m matrix, it's going to return the smaller of the two. Um, so the explained variance of the data here, you can see, okay, principal component one, it explains 72% of the data. Principal component 2 explains 23% of the data. So what this means is you take your data from 4 dimensions and you reduce it to 2 using PCA, you explain almost 95% of your data. So from an efficiency perspective, you've got half the columns of data, so half the data effectively, but 95% of the variance. Um, and if you want to plot the principal components against each other, PC1 versus 2, and then color them in by the species of flower, you can see, okay, we've got these, what is it, Virginica, Setosa, versicolor. These three, you can see they form little clusters. So also you get an idea of like, okay, the PCs also seem to explain some of the structure. Like there's something about the way the data is structured that we can reduce it to 2 dimensions and still maintain the important thing we care about, which is the species of flower. So I know Sozi had mentioned, what if we do PC2 versus 3? PCA values. Let's see. Somewhere in here. Yeah. So we can just change this and say, instead of PCA or PC 1 versus 2, we can do 1 versus 3. So I'm not going to change the labels, but I'll just change the coordinates. And you can see it's a little different. There's still signal there. So what happens when we do 2 versus 3? So those are indices 2 versus 3. These are PCs 3 versus 4. You can see that PCs 3 versus 4 don't really explain a whole lot. And when you plot them against each other, it kind of shows. But, if we go uh, 0 to 1 again, or even 0 to 4, you can see PC1 explains a lot. So these clusters really separate along PC1. So there's different ways to explore your data and to get an idea of like where is the signal actually there. TSNE and UMAP, they're closely related in theory. There's a whole explanation here on the theory behind it. I've also added some documentation. If you are confused by these methods, these are sources I've put together specifically because they're good at explaining it. So this documentation here is really good at explaining it. The UMAP paper is unbelievably dense and full of category theory. Unless you're really interested in like high-level abstract math, I wouldn't recommend it. But there's also a good interactive visualization here that shows how the methods work and how they differ. Does this website still exist? Okay, it still exists. Good. I didn't check that until now. Um, but yeah, let's say we want to work some data here. So PCA, one of the drawbacks is it doesn't do nonlinear structure very well. So this is the Swiss roll data, which exists in 3D. You've got like basically someone rolled something up. PCA is linear, so it might not capture this structure very well. So if we run, this might take a minute to run. Hopefully it's quick. It's always a little touch and go with the online version. If you run this on your computer, by the way, it'll probably take like half a second. It's the fact that it's a remote. It does look like it's taking a minute to run. Luckily, I've pre-generated the figures. Okay, that was not too bad. But if we take a look at how these different methods sort of visualize the data that we have, PCA is on the left, then TSNE, and then UMAP. So with the colors, we can kind of see. Okay, it seems like actually, you know what? Let's get rid of the colors for PCA just to get the point across a little bit better. So PCA, if you run it, you wouldn't really know that this data is actually like a plane that's been rolled up. It's kind of hard to tell because it doesn't preserve that structure very well. A method like UMAP or TSNE, UMAP is a little bit better at preserving continuity. TSNE is a little bit broken up, but you do get the idea that like, okay, it's kind of like patches at least that are connected in some way. So with this toy example, you do get a little bit of intuition as to how these methods work. With the iris data, you can do the exact same as before. So we'll just run what we did before. And this code here, you can change it however you want. But yeah, PCA 1 and 2 for comparison for the iris data. TSNE and UMAP, you can see how they focus on the local structure of the data a lot more than the global structure. So PCA, you can kind of see how it's stretched out in each direction. There's a little bit of continuity between them. But TSNE, it highlights one cluster here and then this one cluster with two subclusters. UMAP also brings up clusters as well, but you can see the sort of overly discretized clustering that comes up at times. So this is something to always keep in mind that it will bring up clusters, but the clusters could be artificial. So those are the toy examples that we have. There's I'm going to not run these notebooks, but I'll quickly go through them. So this is dimension reduction of genotype data. So this is real genotype data from real people in the Thousand Genomes Project using chromosome 22. I've reduced the data so you can work with it rather quickly. But the idea is you can take SNP counts of individuals, and then you can run them through PCA, through UMAP, and through TSNE. And then in the end, you get different visualizations of them. And I've got interactive visualizations. So this is what PCA looks like with the Thousand Genomes data. This is what TSNE looks like, and this is what UMAP looks like. And the reason I've got these notebooks for you is so you can actually kind of compare the different methods and see, okay, like which clusters, which individuals appear or disappear here. So these are interactive HTML. I really strongly encourage you to use to look through them. And then the ScanPy notebook. So this talks about two things. One is the advantage or disadvantage of using a Python package that, say, does TSNE, UMAP, PCA, whatever for you versus the disadvantages. The big advantage is like, if you're pressed for time, you don't super care about like the finer details. A pre-prepared package can do a lot of the heavy lifting for you. So you just plug your data in and then go through it. And the larger libraries do have a lot of community support. Uh, the disadvantage is a lot of people end up treating these like a black box. So I would really encourage you not to do that, but to play around with at least the toy examples notebook that I have so you can get an idea for it. If you do work with gene expression data or population genetic data or anything like that, I do encourage you to work at least once. Start with your data, clean it, processes it, visualize it, start to finish on your own. So that way, when you use these other methods, you have a better idea of what shows up. But yeah, the ScanPy library, it works with PBMC data, and I've got a few pre-run cells for you. So if you just run this from start to finish, it'll regenerate what you have. But essentially, what it does is it gives you a UMAP of your single cell data colored in by the intensity, pardon me, colored in by the intensity of a particular gene, I believe, CD79A, if that makes sense to anyone. I'm not a single cell transcriptome person. I just like to read the literature. And then there's also an example at the end of comparing different expression data and also running clustering. So I believe this is Louvain clustering, or lead-in clustering rather. But just to show um, what you can do with the clustering. So at the bottom of each of these notebooks, there's links that I found helpful. So this one here is specifically for single cell for the genomic data. Server connection error. No way. These are some papers I've written as well as papers from other people that are kind of useful. And then at the intro to the dimensional reduction, I've got some resources. This one was written by my buddy Max, Flattening Mammoths. I think this one is really cool at showing you how a method like UMAP transforms a 3D mammoth skeleton in 2 dimensions. So this shows how the different parameters work. So I would really encourage you to play around with this one, at the very least, just because it's a really cool visualization. Okay, well, it seems like my server doesn't want to work super well anyway, but um, I can stop it there. I can address, I know there was the one question earlier, so let me just switch back to Keynote. Um, Yeah, so that's effectively like a very quick and broad introduction to dimension reduction, visualization, clustering. These are all super deep topics, so you can spend hours on any one of these notebooks. I do hope that you get a chance to play around with them, and shoot me a question if you have it, or just find me at my desk. Some of you know where I am already. In the chat, so there's a question from Chelsea. Would I recommend UMAP over PCA for genetic ancestry in admixed populations rather than for a subset for polygenic scores? And have I worked with clustering in methylation data? So the methylation question, I have not directly worked with it. I have worked with others who have worked with it. And generally what I've seen is they do something like TSNE or UMAP for the clustering or for the visualization, and then often they do the clustering with the lead-in method, usually on the top principal components. Something I've done that other people have not explored is to do density clustering based off of like a 5-dimensional UMAP. So in the references in the intro notebook, my most recent paper from 2026, that one is specifically on genomic data, but I think the method should be able to apply to methylation data as well. In terms of UMAP over PCA for controlling for genetic ancestry, do you mean controlling as in, oh, for polygenic score? So you mean like including PCs in the model versus UMAP coordinates?
Yeah.
Um, yeah, I would stick with PCA. UMAP, I've experimented with UMAP for controlling, for PRS, for GWAS, for things like that. It's much more useful for the clustering, the visualization, and for hypothesis generation. The reason I wouldn't recommend UMAP necessarily, well, there's two reasons. One is that the clusters are not always stable or consistent, so it can work. I know there's a paper from Biobank Japan where they ran polygenic scoring on different UMAP clusters as different populations. So that is a potential thing you could do. I think that that's an area that does need more exploration. But in terms of like including UMAP coordinates as parameters in a polygenic scoring or GWAS model, I would not do that. And there's two reasons for it. One is that the long distances don't matter in UMAP. So there's not really a relation between them. So it doesn't make sense in a linear model. The other reason is that between different runs of UMAP, you're going to have totally different coordinates, most likely.
Okay, that makes sense. I was just curious because, you know, obviously PCA, we can identify the superpopulations and then run a second
Hmm.
PCA within our subpopulation of interest if we're going to do it that way. But I think there's much more conversation around the best way to treat controlling for this and add mixed populations or populate or data with multiple subpopulations included.
Hmm.
And
Yeah, if you're interested in admixed populations and subpopulations, I do recommend you read my most recent paper.
okay.
This is in PLOS Genetics. It's about clustering specifically, but it does talk about how a method like UMAP does pull out clusters of admixed individuals. And my 2019 paper also has a bit about that as well. So those are listed in the references of the intro notebook.
Okay, great. Thank you so much.
Yeah, no problem. All right. Yeah, so we're about at the top of the hour. If anyone has any questions, again, happy to hear them. Otherwise, I believe my email is somewhere. But it's if you just type in Diaz-Papkovich into the brown email window, you will find me.
Thank you.
Yeah, thank you for attending. Thank you for your attention.
Yeah, thank you so much for your time.
All right, everyone. I'll stop sharing there and wish you all a good day and a good weekend.
Thank you, you too.
All right, bye-bye. Yeah, do you need anything from me, Aspen?
Oh, nope, that's everything. Thank you so much.
All right, perfect. Thanks.
Bye.